This work was done by Dr. Ping He's lab in the Department of Cellular and Molecular Physiology at the Penn State College of Medicine. Our research focused on gaining a better understanding of vascular inflammation under disease conditions and identifying prevention strategies. Recently, microparticles, the small cell membrane-derived vesicles released during cell activation, have been recognized as potential biomarkers for disease prognosis. However, the current knowledge of microparticle functions has been mainly concluded from in vitro studies. Their impact on vascular inflammation and disease progression in vivo remains unknown. This study characterized diabetes-induced microparticles and illustrated how they propagate inflammation, contributing to widespread vascular dysfunction. Compared to normal controls, diabetic rats exhibited a more than 100-fold increase in circulating microparticles. The majority of these particles were derived from white blood cells, platelets, and endothelial cells, and form aggregates in circulation. The function of these microparticles were investigated in individually perfused microvessels in rat mesentery with intact surrounding circulation. This approach allows us to quantitatively measure microvessel permeability and microscopically reveal the pathological processes in the perfused microvessels. When diabetic plasma was perfused into a normal vessel or systemically transfused into normal rats, the microparticles immediately adhered to vascular walls, causing leukocyte adhesion, increased microvessel permeability, and elevated vessel susceptibility to additional stimulation. These observations resemble what we observed in microvessels of diabetic rats. Externalized phosphatidylserine on diabetic microparticles was identified as a key mediator of their activity. This study provided in vivo evidence that the elevated microparticles under diabetic conditions actively disseminate inflammation throughout the vascular system. These findings provide mechanistic insight for multi-organ vascular inflammation commonly observed in diabetic patients. Thank you for taking time to learn more about our study published in the Journal of Physiology. The full paper was selected as an editor's pick in volume 597, issue 3. Please contact Pingnian He at pinghe@pennstatehealth.psu.edu if you have any questions.